I'm very pleased that I have an opportunity to talk to people that have different backgrounds. So, having said all that, so this is my opportunity to have some pretty pictures in my presentation. So I've taken it. So I wanted to briefly introduce the settings, where do we work and what do we do there. So the Oxford Genomics Centre is effectively a collaboration between the high throughput sequencing, which high throughput genomics, which I'm part of, and the bioinformatics course. So two different entities, independent, but there's obviously a lot of interaction that is required to deliver results and products to our users. So it's a core facility. We are a service provider for people internal to the department, the university, and we have collaborations and work done with other universities and other countries as well. So what we do in high throughput genomics can be divided into three main areas. So we have an array team who's dealing with Illumina and Affymetrix arrays, and we also have an Agena platform for genotyping. Given my role, which is basically develop, test, evaluate, implement new applications, I have very little to do with these guys because pretty much everything that is array related seems to be ready off the shelf. You plug it in and run with it. I'm not entirely sure that that's true, but I'm happy to live with that. So I'm busy enough as I am. Completely on the opposite side of the spectrum is the single cell platform. So we've been in this arena for a little while, probably in a year and a half or so. And so as Dave mentioned, we have a C1, we also have a Polaris, and we have a cell sorter. The idea was to set up a facility within the Wellcome Trust, stems from an initiative in Oxford that developed a consortium. So it's multiple departments that have been involved with it. So there's been a big grant awarded to the consortium to buy equipment. So you buy the usual stuff. And also there was a bit of a lack of sorting capabilities within the center. So we opted for a Sony instrument which promises to be very user-friendly. So the model we have in mind is a walk-up model, so where people can have their crazy hours and just do the experiments and run their own cells without bothering us too much. But I'm not going to talk about single cell much more than I've just done. And finally, there's the general, the big component of the group is everything that goes and feeds the Illumina machine. So we have a few sequencers, and we have been doing NGS on Illumina machines for quite a few years. People know us in Oxford, and so we have a lot of work in this area. So this is an attempt to give a sense of what sort of involvement we have. So this shows about 200 unique customers in a single year that we had, spanning across 700 different projects, and for a total of about 20,000 samples for sequencing and almost the same for arrays. This is, compared to Sanger, it's not big numbers, but the thing to superimpose to these numbers is the fact that these are not all same workflows or high throughput projects. So we get a variety of applications and weird questions from people that keep us interested and motivated. And a lot of projects have very low throughput, so just a few samples, and occasionally they need some custom adjustment and tweaking to get to the desired results. So, it's a lot of work, and we keep busy. And to give it, even more of a sense of what we've been involved in terms of adapting existing technologies and solutions to the requirements from our users and customers and collaborators, I'm following almost subconsciously the pathogen resequencing theme to illustrate a couple of examples. So we've been developing or adapting, I should say, an RNA-seq protocol to be able to detect and sequence full viral genomes from pathogens. And more recently, we added an additional module to this protocol, which allows us to capture the reads of a desired pathogen and enriching for them and reducing the requirements for sequencing and therefore costs. And this illustrates this plot shows the viral load as determined by an independent clinical lab and the number of reads detected after sequencing, and shows that if you do a metagenomic sequencing, which is hardly surprising, you get a number of reads which is proportional to the viral load in a fairly decent manner. What was surprisingly, and we were very pleased to see, is that the same correlation can be maintained even after capture. So not only we can reduce the cost by sequencing the same number of samples on a MiSeq rather than a HiSeq, but we also maintain a piece of information which is quite useful to either QC the samples, make sure that they're the right thing, but also potentially provide an alternative ways to determine viral loads in clinical samples. Another push that we have from pretty much all directions is also to quickly implement new and promising technologies. And this is an older case where we just purchased a MiSeq in those days, and we wanted to test how this instrument could impact the detection and evaluation of pathogens in a clinical ward. So what we did was to collect some C. difficile samples, where we received them, I should say, and we did the sequencing from start to finish. We wanted to test how long does it take in a realistic workflow. And we managed to deliver the data from sample to data within a week. And in those days was probably a couple of months to get to the same level. So it was a big impact. And the fact that MiSeq has been adopted in many clinical labs is also a confirmation that this is the speed is achievable and is quite important. So more recently, we've been using an alternative sequencing technology, which again has many promises. I don't know if you know about Oxford nanopore sequencing. I won't go into too much details, but if you're interested, I'm happy to talk again afterwards. So the main take-home message from this complicated figure is that instead of sequencing your DNA by synthesis, as on an Illumina machine, here you just detect the native DNA, if you want, as it goes through a pore. The pore is just big enough for a single molecule, a single strand, I should say, of DNA to go through. And the current potential difference is measured as the DNA goes through. And so you get this kind of profiles. And you notice that the pore is bigger than a single nucleotide. So what you're measuring is, in fact, an oligomer going through the pore. So that complicates a little bit things when it comes to base calling, but algorithms are getting better and better. And so long story short, this is a quite exciting technology, allows you to do very long reads comparable to PacBio, but instead of having a fairly large platform, you have something that is it's about this size. So it's very portable. You need a laptop to connect to for the data acquisition and crunching. And it's so portable that, I don't know if you noticed, but a couple of days ago, it's being released on bioRxiv, the publication of a paper by the NASA people who've been sequencing in space. So I think that's as portable as it gets in these days. So what have we done with it? So again, we supported a team who's been developing methods to rapidly detect resistances of pathogens to known antibiotics. And the implementation of a nanopore sequencer allows point-of-care type of transition. So this can empower hospitals and GPs eventually to do tests very rapidly in place without sophisticated facilities, which goes against my personal interest in a way, but I have other plans, so I'm happy with that. The bottom line here is that nanopore, despite the error rates and everything that you can think about it, was still good enough to recapitulate the predictions from Illumina sequencing, with the advantage that you can do that more quickly as well. So one of the things about nanopore is that very soon after the DNA has gone through the pore, you can get the sequence information from that single molecule, which means that you can keep monitoring the data and stop as soon as you have the information you wanted. So that can go down to just a few hours in some cases. So I'm not sure I convinced you that we do a lot of very different things, but hopefully that's got through. So how can we envisage the implementation of a new instrument in such a disjointed almost a scenario? I hope my boss doesn't see this. Can we edit this part afterwards? So the way I approached this was to have a look at the workflows and trying to summarize the points that are in common to everything that we do. So no matter what, we pretty much QC every single sample that comes in, unless it's a single cell. So it's kind of tough. And we use different methodologies to do the QC. So picogreen, tape station, gel, and nanodrop. We heard of some of these, if not all, already. And picogreen has already been shown as amenable for implementation and use of echo to feed. So that's your first candidate. Sample preparation. This is very hard to summarize because at the moment we have over 15 library preps that we offer as more or less standard, plus all the variation you can think on top of that. So I would spend a little bit more on library preparation in a few slides. And finally, we need to have a library QC. We need to know if the library is worth sequencing, because the sequencing cost is quite important, it's quite high. And again, we can use picogreen, tape station, and qPCR, and now we have two elements that can feed into an echo context. And I'm very pleased that I predicted right that picogreen and qPCR were going to be covered by other people, so I'm not talking about those. So, how do you bring an echo into genomics? Sorry for, I had to have a jokey slide. I usually have sci-fi slides in my presentations. I resisted that, but something is wrong with me, I guess. So, as I said, we got some money to spend on instruments a little while ago, and we looked at different liquid handling solutions. We looked at mantis and mosquitoes and other things. The echo was my preferred option. I pushed a little bit for it, although it's more expensive than others. To me, gave more flexibility than any of the other platforms that we tested. And if I had to summarize the major point that you would make to buy an instrument like that is that, as we heard before, it's fast dispensing for small volumes, so you can scale down reactions. There's no tips involved, so you save on consumables, you save the environment, and you limit cross-contaminations, if not avoiding them completely. And it allows you to more easily go to higher density plate format, so 384 is a lot more feasible than on conventional liquid handling. So, having said all that, I'm going to show, the first example I'm going to show, it goes against everything I've just said. So here is an exercise that we did with a PhD student who was visiting the lab. And his interest is in pre-implantation diagnostics. So we wanted to see whether we can do whole genome sequencing and amplification and sequencing starting from a small number of cells, ideally 1, but 3 to 5 is fine. So, and also it's a few samples. So it's not high throughput, it's just one or few samples. And 529 and kits that do whole genome amplification are fairly cheap. And because you only do 1 samples or so, reducing cost is not really a major target. So why using the echo? So the reason why I thought it was a good idea is because there's another feature of the echo which allows you to design very rapidly, as we heard before, several iterations of the same reaction. So you can test volumes, you can test conditions, ratios of enzymes and buffers fairly rapidly and in a single or few runs. So we took advantage of that. And we tested a couple of commercially available 529-based kits. One is from QIAGEN, and the other one is from Cygnus. And after determining the ideal conditions in terms of reaction volumes that we were happy with, we tested the kinetics of amplification. And the idea here is that we wanted to limit the amplification to avoid some of the known artifacts that come with this technology, and to a point where we can reliably generate enough DNA that is compatible with a sensitive library prep. So, and obviously this also cuts the time that goes from sample to data. So we identified that within a couple of hours, you quite reliably get enough DNA to feed a library prep. And the library prep of choice for this workflow is, unsurprisingly, Nextira. As we heard a couple of times already, this is a ligation-free, let's call it, library preparation, which takes advantage of a transposase conjugated to adapter sequences, and in just a matter of minutes, it can fragment the DNA and ligate adapters at the ends. So, you get a few steps condensed in just a few minutes. And because of these features, it can work with very low amounts of DNA. So it all sounds like the right combination. So what we wanted to do was also test how important it would be to calibrate the amount of DNA that goes into each reaction. So because of the way it works, transposase needs to attack the DNA. It's kind of intuitive, if not already published, that the ratio between the DNA and enzyme is crucial to get reproducible libraries. But nevertheless, we wanted to see, in this particular case, how bad the situation would be. So we chose 3 samples, one of which was of higher concentration. And you can immediately see that there's a difference. However, if you were in a rush and, for whatever reason you decided so, you could get away with this. So this is, in fact, a sequenceable library, which is good because it means that you have an option, if you were brave enough, to skip at least a couple of steps and go directly into sequencing. However, to have a more generally applicable workflow, we decided to add a step, which is unsurprisingly quantification and normalization. So again, after the reaction, we can take an aliquot of the amplified DNA and then normalize the reaction accordingly. And these are the same 3 samples that I showed before. After the normalization, you can get pretty much overlapping profiles on a tape station. And I'm showing this slide because there's a couple of things here. One is that we bypassed a cleanup step that is actually essential between whole genome amplification and Nextira library prep. If you just go straight into library prep, that fails miserably. But we didn't want to have cleanups not to lose material. So that's one thing. The other thing is that we didn't dilute the DNA to get to these results. We actually added more or less enzyme. And this was easily achievable because we delivered the enzyme with the echo. So because this is not RNA, so amount of material is unlikely to impact in an unpredictable way the results. We are happy to use as much of the DNA we can within reason in the prep, and we can do a decent job by adjusting the amount of enzyme we use. So in this case, what we did is, because costs are not necessarily a big issue since we're dealing with one or few samples at a time, we decided to take the whole genome amplified material, dilute it down so that we have enough to do any QC step that we like, but also by doing so, then use everything that is remaining and go straight into a larger volume Next-era reaction. And just by simple means of diluting the sample, that was enough to get from amplified DNA straight into library prep without cleanups in between. So we're kind of happy with that. So one of the funny things that happened pretty much even during the demo of the echo is that having so many workflows there and so many exciting opportunities to work with this machine, we said, okay, let's pick something like 5 different protocols, do a quick test, see what works and what doesn't, and we just pursue what works and leave what doesn't for troubleshooting later on. Unfortunately, what happened is that everything pretty much worked. So we got to the uncomfortable position of having, how do we choose now what to do? So instead of choosing something, we went, okay, if everything works of what we tested, how about testing more laborious and more sophisticated workflows that are on a single step protocol? So we went back to a more traditional library prep method, which is depicted here as multiple steps. In the old days, at the end of every step, there was a cleanup with either columns or beads. Now kits have been developed that allow you to simply add reagents to your previous mix. And some kits still have a cleanup, usually after ligation before going into PCR. So we say, okay, does this still work? And the reason why I'm particularly interested in testing these is for 2 reasons. One, it would provide a case to demonstrate that you can do more complicated workflows, so when you can keep adding reactions to it, and SmartSeq2 is an obvious member of this category of workflows, if you like. And the other thing is that this protocol is sometimes better suited for bacterial genomes than Next-era, which is very sensitive to concentrations and quality of DNA. So if you get large-scale batches of samples extracted by other people with mixture of different genomes, it can be a nightmare to resolve it with just Next-era. So there's a few good reasons behind this choice. So sorry for the busy slide. So this panel first. So the bars illustrate insert sizes as detected after sequencing from library generated using that workflow using kit 1, which I wanted to keep private, if you like, simply because we butchered the kits. So we got some dodgy results here and there, and I don't want that to be associated with the name. So this is in fact an NEB kit, but bear in mind that whatever you don't like about this is not the kit because we didn't follow the manufacturer recommendations. So what is striking here is that despite the fact that this DNA was all fragmented on a sonicator, on a covariance machine, so all the DNA was actually the same size, after sequencing we see a shift in sizes. And the shift is associated with the treatment that we chose to apply to go from ligation to PCR. So in this case, we just simply hit inactivated the ligase, just to prevent possible artifacts while incubating the master mix before PCR, and just use the next add the PCR mix afterwards, and as compared to a more canonical cleanup step in between. So this is the first and probably the most concerning alarm bell there. In terms of percentage of duplicates, everything is below 1%, so I'm happy with these numbers. Yields are variable, but this is not necessarily a library prep issue. It's, there's a pooling step that goes afterwards, and slightly imprecisions there can generate variations there, and even considering that the difference in number of reads is not huge. The reason why I have the yields there is because it helps a little bit with the mapped reads. It's easy to have a high percentage of mapped reads if you have only a few reads sometimes. So this gives a little bit of a framework for it. So again, the mapping is quite good. You could argue that it's slightly better for the cleanup, but I wouldn't go necessarily that far. So what the hell happened with these guys? So we got shorter libraries after sequencing them, the DNA that we generated. So we had a little bit of a closer look. We looked at GC biases, which is again a very useful metrics for this type of applications. In case you're not familiar with this type of plots, the red bars illustrate the frequencies of 100 base windows of the reference genome according to arranged by GC content. So this goes from 10 to 80, which is the E. coli genome range, and you can see that there's the majority of the genome and fragments are around 50%. Yep. And on these other axes, we have a normalized coverage on GC. So in an ideal library, you get every single point aligning on this line at 1. So this is a little bit off. This is actually horrible. This is one of the worst libraries I've ever seen in my career, and I've seen a few. On the other end, we have these. So by simply having the cleanup that we so desperately wanted to remove, we got probably the best data we've ever seen with this type of kits, considering that this is a PCR library. So similar plots are usually obtained if you do a PCR free. So you remove the bias introduced by the PCR step itself. So that was a very surprising finding, and we're very happy with these results. So we're planning to do a little bit more evaluations of different GC content genomes and so on and so forth. But I still have the obsession of removing stuff that cannot go on the echo as much as I can. So the two steps that are still here that we had to do offline. One is this sonication, and one is this cleanup. So we had a look at a different kit, which I'm going to talk in a couple of slides. Sorry, I always forget that I have these slides first. In trying to understand what the GC bias meant for this data set, we had a look at the coverage across the genome. Simply because one possibility is that you have a region of the genome which is particularly affected that you don't recover, and everything else is absolutely perfect. And in some circumstances, you can think that it's good enough for what you want to do. And as you can see, this is not the case. So the blue line shows the hit-inactivated library, and the red one is the cleanup library, and you can see how much tighter the coverage distribution is after cleanup. So again, that means that if you wanted to skip that single step, we will have to sequence a lot more of the DNA and the library to get to the same average coverage, which is not a very good idea. And this is a blown up of the same plot showing that, interestingly, and probably not so surprisingly, you do have troughs that are present in both library preps. So there are regions that are difficult to sequence or to amplify. And but overall, you get much more spikiness if you hit inactivate simply. So this is mystery kit number 2, which is kappa equivalent. Again, whatever you see is due to the fact that we use it in specific ways, and disclaimer. So the advantage of the kappa kit is that in the kappa hyper plus version of it, you have an opportunity to do fragmentation with an enzyme mix. So immediately you can think that, okay, I can transfer the enzyme using the echo, and so I can bypass the sonication step. So what we did here is to test the enzyme fragmentation followed by cleanup, heat inactivation, or replace the enzyme fragmentation with Covaris as before to compare the two data sets a little bit better. As you can see, the insert sizes vary, and this is a big concern. I'm going to spend a little bit more talking about what's happening with the fragmentation with using enzymes, which is a little bit of a headache at the moment. But anyway, as before, duplication rates are not too bad. This was done on a different run, on a smaller scale, so the yields are lower. I apologize for that. The percentage mapping is still quite decent, and you can immediately see that we have some libraries that are showing some problems. So we didn't get as many reads as expected. So we knew that from the QC that the libraries were very weak. We tried to see what we could get out of it anyway, and all we could tell is that this is the right genome because it maps, but the yield is very disappointing. So, and then we had a look, as before, at the GC content. I said, oh dear, this is far from being a straight line, and this is, forget what I said before. This is the worst GC plot I've ever seen. Interestingly enough, this shows a position effect that we identified when dispensing the enzyme to the samples on the plate. So remember, this is a 384 well plate. We simulated the time by dispensing water in between, but the difference is that these are the so these are the top corners. So these are the samples that received the enzyme first, and these are the samples that received the enzyme last, which is slightly counterintuitive. But what we think is happening is that the enzyme is very reactive, it's very quick. So a lot of the reaction, the chopping up of the DNA happens during the dispensing on the echo, even at room temperature. And this means that these samples didn't get to the same level of fragmentation, which is good enough for the other samples, and somehow impacted on yields and GC content and all these things. So we are thinking hard of ways that we can bypass this problem, and I'm not sure that there is an easy solution. Probably we can just resign to the idea of using a sonicator outside of the echo and just go for the best results ever seen. So similarly, this is the heat inactivation profiles now. So again, we see that positional effect, which is very annoying, but we don't see the same effect that we saw before with the heat inactivation. So this kit is a better kit in terms of skipping cleanups. In fact, I believe that the new version of this kit now has removed the cleanup step between ligation and PCR. So I wish I knew that before we did all these, but hey. Still not perfect, a low GC, there's a bit of bias. That's probably okay because we tend to have either genomes around 50 or higher GC content. So this could be a decent workflow for that. But is the problem really the fragmentation, or is there something else going on? So that's why we did the covariates fragmentation as well. And that shows that, yet a different profile, but a lot better. So if you do fragment without the enzymes, the 4 replicates actually are now quite nicely behaving in the same way. And so this is potentially a solution to remove the cleanup step. So we have 2 options at the moment, which we're trying to potentially combine. So if you could combine the 2 kits and get the best of the 2 worlds, I would be very happy. But we have a solution which potentially allows us to remove the cleanup and get some acceptable data. Possibly we could remove the PCR altogether and see whether that improves even further, although part of the drive for this type of application is to reduce the amount of samples that goes in. And this is quite important for pathogens that grow very slowly, like TB. It takes weeks and weeks. So if you can reduce the time from sampling to DNA that can be sequenced, that would be quite useful to the clinical community. So we have these two kits which we're trying to figure out how to combine eventually. But in the short term, I think we do have a solution, which is the kit I showed before. Even if we have to do a cleanup, that's probably not the end of the world. We've seen other people having similar problems, and you can take the plate offline. We do have, luckily, quite a lot of conventional liquid handling that we can implement to do these cleanups. So to wrap it up, I guess, I hope I gave you a sense of how we see this instrument. And I think we are the only group without an access. Is it? So our integration is manual, so we don't need someone. Okay, so we may have to work on that. So just for peer pressure. So we see a lot of opportunities to implement and slot in an echo into our workflows, and that's very good, because when you recommend an expensive instrument and it doesn't deliver what you promised it was going to, you get in trouble. So once again, I think I got away with it. We can also take advantage of the flexibility and the ease of programming the instrument to quickly assess optimizations that we want to develop, but also potentially support better custom protocols that people constantly request. Cleanups are still a little bit of a bug in my head. I'd really like to find a solution and remove them completely. I realize it's not an easy task, but again, this I think would be a perfect environment to discuss ideas and opinions. So if anybody is interested, either now or coffee or later on, please get in touch. And I feel like everybody's saying beautiful things about the echo, so I wanted to again break the mold slightly and have more than a complaint, a request. So we get a lot of high-profile visitors that know very little about genomics but have loads of money. So funding bodies and sponsors of some sorts that they get a tour of the lab, and we show all these lights moving on the sequencer. That's good enough. Then we have liquid handling moving things around, and they get all very happy with that. Then you get to the echo and say, this is a fantastic instrument, and you can't show anything. So can we have a see-through box version of it so we can show that it's actually doing some amazing stuff inside? Just on the wish list. Yeah, okay, good.
I'm taking notes.
Perfect. So finally, I just want to acknowledge the core people that actually helped a lot in generating the data that I've shown today. So David is the head of Untitled Genomics. The staff of Maria Teresa and Amy did a lot of work trying to break the system somehow and getting fantastic results. I've already showed you some of it. Araz has been is the guy who developed the workflow for whole genome amplification and library prep. Lorne manages the sequencing machine team, and I had to put it there because it has to deal with a lot of weird stuff that we generate, and he needs to sequence. And equally, John, who manages the pipeline, has been quite reasonably tolerant with adjusting the pipeline to accommodate with the weirdness that we throw at him. So, with that, questions?